Thank you so much, Robert, and thank you indeed for the invitation, and to you and your colleagues. We're really delighted to be here. You can control the slides, Matthew. So we'd like to begin by acknowledging the traditional custodians of the land on which we stand, the Turrbal peoples from the north side of the river, and from the south side of the river, the Yagara peoples. And we pay our respects to elders past, present and emerging, and extend this respect to any Aboriginal and Torres Strait Islander colleagues in the room and online today. And we've brought a little bit of Darug country in Western Sydney here today through the artwork of Leanne Tobin. And Leanne's artwork reflects the vibrancy and traditional motions of the dragonflies as they move through their journey of life, which is quite lovely, I think. Thank you. We'd like to acknowledge our team. So we'll be drawing on a few studies today, some of which we've been involved in, and here are our fantastic collaborators, and we'd like to acknowledge them throughout the presentation as well. So you go ahead, Matthew.
Yeah, so if you're able to go to menti.com and type in the code on the screen here, there'll be 4 questions, 0 to 10 being 0 being least knowledgeable or confident, 10 being the most. You can just fill that out. That should come up on live. So the code is 45654109. Very interesting. All right.
Give another minute.
Yeah. Okay, looks like we're pretty much there. All right, excellent. So we'll do a post scale as well, just to see if we've done our job.
We'll do our best. Thank you, that's really helpful. So in terms of the learning objectives today, by the end of this session, we'd like you to be able to describe microarray patches and understand their role in vaccine delivery, understand the evidence to date about microarray patches for use in vaccination, identify factors that influence public health and healthcare professionals' acceptance of microarray patches, explain the features of microarray patches that enhance usability for healthcare professionals and for the patient, and also to identify the barriers and facilitators for microarray patch use in clinical settings compared with use at home. So just broadly, why are we discussing microarray patches? So the Vaccine Innovation Prioritisation Strategy, which is a consortium including the World Health Organization, UNICEF and Gavi, recently ranked the development of microarray patches for vaccination as the highest global priority for achieving equity of vaccine coverage, and particularly in low and middle income countries. And within Australia, I'm sure many of you have probably seen this too. I recently submitted some feedback on a consultation paper, which is called Towards Australia's National Immunisation Strategy for 2025 to 2030. And in that consultation paper is mentioned the following. So technological advances may require significant adaptation in delivering immunisation programs, and will always require deliberate efforts to build trust in new technologies. And we know that can be an issue in terms of communicating that to the general public in particular, and particularly during COVID with the mRNA vaccines, and communication with key stakeholders, including consumers, employers, community and clinical groups, will continue to be essential to build confidence in new technologies. And in terms of opportunities for action in the consultation paper, preparing for assessment of emerging vaccine technologies, and for the opportunities and challenges that adopting new technologies may present to existing immunisation programs and delivery. So we're sort of addressing some of that today. And obviously, this is really important to Sustainable Development Goal number 3. So microarray patches are a disruptive technology and an innovation that can improve accessibility and implementation of vaccine services globally. They consist of tens to thousands of microprojections applied to the skin by finger pressure or an applicator. And I've actually got, this is the Vaxis Have many people seen these? This is the Vaxxis microarray patch, the Generation 3 one that I've got sort of for show and tell at the moment. And we've got sort of a demonstration video that we can show you soon as well. So they penetrate the stratum corneum and deliver vaccine to the epidermal and upper dermal layer, which of course are rich in antigen-presenting cells. And the dermal dendritic cells play a key role in intracutaneous vaccination due to their ability to activate an adaptive immune response, which is the role of adjuvants, as we know from our earlier speaker, in commonly administered intramuscular vaccines. There are several types of microarray patches that are currently in preclinical and clinical trials, including the biodegradable or dissolvable coated, solid, hollow, hydrogel forming, porous and hybrid microarray patches. And for each of these designs, applying pressure to the map enables the projections to penetrate the outermost layer of the skin with less pain than a traditional needle and syringe injection.
So the reason why dissolvable and solid were in bold is because they are the most commonly used in clinical practice at clinical trials at the moment. So the solid is typically made of a polymer or a metal and is coated in the antigen, which is then delivered to the skin. These polymers, they stay intact as they exit the skin and are then disposed of. Alternatively, there's a dissolving patch, which are made of biodegradable polymers. These dissolve in the skin. Yeah, so hopefully we've got some nice examples here. So on the very left is what we just saw with the Vaxxis applicator. So that is a slightly older generation. However, at the top there is the actual HD map, the actual patch itself, and below that is the applicator. Now in the middle here we have Micron. So a similar thing. We've got the dissolving patch with this one at the top there, and below hand you can see that the applicator is just there. Now on the right there's a prototype from Queen's University, Belfast, which is another type of dissolving map.
Just this, a bit of an overview of the kinds of studies that are currently taking place at the moment, and this is a summary from PATH. And currently there are teams evaluating the cost of manufacturing and delivering vaccines via microarray patch. There are teams conducting economic evaluations with microarray patches compared with delivery via standard methods. There are also teams conducting design and human factor evaluations to assess the usability and acceptability and fitness of maps for vaccine programs. There are teams testing the thermostability of microarray patches to understand potential storage requirements, and also teams conducting clinical research to compare the safety, efficacy and acceptability of microarray patch delivery with standard delivery methods. And in terms of paving the pathway for introduction, identifying and facilitating product development pathways for microarray patches, including the relevant clinical, regulatory, manufacturing and program introduction activities are very important, as is engagement with country and regional stakeholders to understand decision-making considerations to pay for vaccines via microarray patches based on proposed product attributes. So in terms of some of the benefits and limitations of microarray patch, so there's a reduced pain and discomfort score, which is very important for consumers, ease of use. There's equivalent or improved immune response with delivery via microarray patch. Obviously, there's reduced sharps risks with no needles. There's certainly enhanced thermostability. There's a potential for reduced healthcare associated costs because a cold chain is not required. There's reduced clinical waste. There's potential for trained lay people to administer the patch. And one of the most innovative things, of course, is potential for self-administration by patients, and also mass administration. So there could be supervised self-administration within a mass vaccination setting. In terms of the limitations, there's limited vaccine compatibility and antigen loading. There's complex manufacturing. localized skin reactions such as erythema, the cost of development, which is substantial. There are technical limitations, and most of these are tied directly to regulatory approval, and obviously it's currently not approved for human use. We're still in sort of phase 1 to clinical trials.
Yeah, and so to understand a bit about what we're talking about in terms of safety, usability, acceptability, we've taken some definitions from the TGA and the International Organization for Standardization. So safety is described as a product's ability to eliminate or reduce the risk of potential hazards for intended use or foreseeable misuse. This includes adverse events following immunization. The usability refers to the extent that a product can be used to achieve its desired goal effectively, efficiently, and satisfactorily by users. Acceptability is the extent to which an intervention is received by population and meets the population's and organizational settings needs. So here we have some adverse events from some of the phase 1 and phase 2 trials that were conducted. So we've got 4 or 5 now influenza studies, 2 in measles and rubella and 1 in Japanese encephalitis. So systemic adverse events were not common. The less common ones were fever and headache. People rarely experience fatigue, cough, sore throat, diarrhea, myalgia. Local adverse events were common with mild induration or erythema. Less commonly, discoloration, exfoliation, site pain, pruritus, and pigmentation. Now, in one of the MR studies, there was a papular rash, but that was rare. Now, on the bottom row there, we have the placebo type studies. So systemic adverse events were quite rare. However, in one study, they did report diarrhea, vomiting, nausea, and headache. Again, commonly erythema was present, and less common tenderness, purpura, edema, and petechiae. Now, this is a real picture of me not a few weeks ago battling COVID for the second time. So immunogenicity was studied in these phase 1 and 2 trials, demonstrating equivalent or even stronger immune response or immunogenicity compared to injection. Very few studies have investigated the specific pathways. However, they shared similar immune responses. And these studies were conducted in healthy adults aged 18 to 57 for the influenza, Japanese encephalitis, and MR vaccines. One study with investigating MR did it in infants and toddlers. Now, these are some examples, again, using dissolvable and solid maps. So you can see that you don't have to worry too much. You can take a photo if you want. But these have the immunogenicity, the geometric mean titers in the second column here. So you can see that they are quite similar, but in fact, sometimes the map was higher, whether that's statistically significant or not. And we also have the seroprotection on the right. So you can see that essentially it is equivalent, or in some cases, it did provoke a stronger immune response. Interestingly, the Japanese encephalitis one at the bottom there was a low and high dose in that study, and the low dose performed just as well as the injection.
So in terms of acceptability across all of the studies that have been conducted thus far, participants preferred microarray patch over needle and syringe in 4 studies. Many healthcare professionals wanted microarray patches in their clinical practice and expressed willingness to administer. All the points with an asterisk are from studies that we have led. Many unvaccinated individuals would be willing to receive an influenza vaccine via microarray patch. Microarray patches were considered more convenient than a needle and syringe, and that's by both healthcare professionals and lay people, patients. Self-administration was more acceptable in clinical settings. Trained lay administration was viewed favourably, but healthcare professionals were more cautious, unsurprisingly. And wear times of less than 30 seconds were acceptable. Participants reported that microarray patches would be useful in children and needle-phobic clients, adolescents as well. And there's huge potential for mass administration. So with regard to the settings and acceptability, so in clinical settings, there's the microarray patch is highly acceptable among the general public and healthcare professionals. Healthcare professional administration was highly acceptable. Trained lay people or community health volunteers, it was highly acceptable to them as well. Obviously highly acceptable in mass administration, and there's reduced clinical waste, which was regarded favourably by all. At home, in terms of self-administration, there were concerns about lack of access to medical support. There's concerns about documentation, for example, on the immunisation register, and concerns about disposing clinical waste. And in terms of cost, there's reduced costs with obviously with regard to cold chain management. There are concerns about reimbursement and coverage, particularly in health systems where it might, in the US, for example, and there are concerns for low income earners if they had to pay, a small fee, for example.
So in our studies, we've found that the general public and the healthcare professionals found maps to be easy to use, particularly with illustrative instructions for use. And some have also noted that the video demonstration beforehand would be particularly useful. In our older adult study, we did note that there might be issues applying the device, removing the foil, that kind of thing. However, they did do very well, including those with dexterity issues. Wear times do differ quite a lot between the different devices, with most from 10 seconds up to 20 minutes, and in some extreme cases of hours. In one of our studies, a visual indicator was suggested by general public to give them a bit more confidence. However, it was not suggested or recommended by the healthcare professionals. Most felt that it would be easy to administer to children. However, there were some healthcare professionals thinking about the more of the toddler situation, Wrigley toddler, where they would prefer a shorter wear time. About 10 seconds would be more acceptable. Anything over 30 seconds might be a bit challenging in those types of situations. So PATH, which is an organisation, you might have heard about it, in terms of vaccination and microarray patch research. So some of the target vaccines, in addition to what we've discussed, include rabies, post-exposure prophylaxis, COVID-19, human papillomavirus, and the birth dose for hepatitis B. Some other priority areas are long-acting HIV PrEP or pre-exposure prophylaxis and contraception.
So the microarray patch regulatory working group aims to inform, guide and define the regulatory science of the microarray patch dosage form. Obviously, there's a lot of work to be done in this area, and the group aims to expedite the clinical translation of the technology to benefit key stakeholders, including product developers, regulatory authorities, public health bodies and end users. And I won't go through those 4 streams of work, but this is what they're currently working on. And it's quite complex because obviously you've got to do FDA, European regulation, TGA, and they're all quite different and they're all at different stages, and they classify the microarray patches and applicators differently as well. So lots of complexity there. And we'll just briefly summarise some of the studies from our team so you can sort of see what we're involved in and what's happening next. So we conducted a usability study with healthcare professionals and professional immunisers, and this was within an Australian setting. It was a feasibility study. We use basically, it was qualitative and quantitative mixed methods, and also observational as well. And I'll just move on just in terms of, I guess there's some of the key findings from this is that what we observed is that the more qualified the professional immuniser, so a doctor or whatever, versus a healthcare worker, the least training, how can I say this carefully? The least training, the more compliant the person was at using the microarray patch on a patient, and the more experienced the person was, the more they assumed they thought they knew how to do it, even though it was terribly sort of appeared very straightforward. And that was basically with regard to adherence with the hold time or the wear time on the patient. So there's some key learnings from that. But look, it was highly usable and highly acceptable to everyone. We'll just move on to the next study, and you can summarise it.
So this is a piece from my PhD research. So this was a sub-study of a project to validate a scale to measure MAPS safety, usability and acceptability. So we conducted surveys across Australia, Canada, the United Kingdom and New Zealand, and we did interviews within Australia. So I should mention, before they attended their survey or their interview, they did watch the video demonstration that I was trying to show you earlier on. And they read the instructions for use. So overall, we found that the general public and healthcare professionals shared similar ideas, that it was safe and effective, that it was easy to use and acceptable. In our interviews, we discovered that healthcare professionals were interested in incorporating MAPS into their practice, but were concerned if there was a potential for home administration without supervision in case of that anaphylaxis. Now, most of them felt it was straightforward with those instructions, and all discussed the advantages for children and needle-phobic individuals. So we'll conclude by discussing the potential benefits and the challenges going forward. So we have discussed that MAPS have been immunogenic, well tolerated, easy to use and acceptable. There is a potential for reduced burden on healthcare professionals and its resources, with the potential of lay and self-administration and other time-saving factors, such as not having to order vaccines or reconstitute. There is convenience and mass administration with a role in potential future endemics or pandemics. And of course, there's reduced pain and enhanced thermostability. Now, some of the challenges is the safety of unsupervised self-administration, the accuracy of any documentation. So how would that get documented on air, for example? Potential issues with confidence in new technology, as we've seen that people are hesitant, as we've seen some of that with the mRNA type vaccines. And lastly, there's an unclear time frame to regulatory approval at this stage. Thank you very much. If we could pop back, don't use that QR code, I don't think it's working. You can just go back into your menti.com.
And then while we're doing that, if we do have 30 seconds or whatever, I don't know if our colleagues in AV are able to show that video at the end. That would be fabulous, I think. Hang on. Well, watch this and then we'll come back to the question.
Yeah, that's fine.
Are there plans for clinical trials with influenza vaccine for children? People are noticing that the uptake of flu vaccine here in Queensland and across Australia is well below what we would hope at 25% or less. So are there plans for clinical trials using MAPS for flu vaccine in children?
So there's there'll be another study with adults that's moving into a phase 2. And there is plan for a paediatric placebo trial before going to vaccine. So yeah, so we're involved in that. So obviously we want to do very similar to the kind of adult study that we shared with you to make sure that it works, that the patch actually works effectively in the skin of children. But then we would move to antigen. But yeah, I think I totally agree with that comment. Thanks, Robert.
So I'll cut off the online questions, sorry, but questions in the room. Quickly,
Michael
please.
Murray, pragmatic geriatrician. I don't think old age is going to be a problem at all because we've been using patches on them for years, probably too many, if anything. So patches are easy for wriggly people. We all use between the scapula and the spine, and it's quite hard for even the most dexterous to remove anything there. So that shouldn't be a problem either. So the bit you've been tantalising me for years is, when are we going to get some?
Yeah,
Because delivery is a real hassle. We've got 749 facilities in Victoria. So just delivering to those environments, let alone at-home community, is really pragmatically problematic. So I just want the product.
yeah, don't we all? I'm with you on that one as well. So yes, I think the regulatory approval, I went to a conference, a microneedle conference in Seattle last year. I think the really exciting news is actually that we're having a microneedles conference here next year, the 11th to the 13th of May. Put it in your diaries. Come. It'll be in Brisbane. UQ won the bid and I'll be on the scientific committee. So really, really exciting. I think the regulatory approval is very complex. One of the presentations I heard at the conference last year was one of these startup companies failing regulatory approval. So I think there's plans that Matthew was talking to me about, actually, about a consortium of companies going in together and going for regulatory approval. And of course, it's all the different, it's very complex. Yeah, but I agree.
We're over time, Michael, so keep it brief.
Very quickly, I also have an interest in wound care, and I understand that very well that the older person's skin is very different to younger people's skin. Are we sure that a microarray is going to work as well in thinner, drier, etc. skin?
Yep, that's exactly the study that we've just done with placebo, that is. So and yeah, so with placebo, it was absolutely fantastic and really great results. So I'm about to finish that manuscript when all these conferences are over, but I've got about a day's work on it and then it will come out. And yeah, we were very concerned about that. And so we're really happy to see the results. Thank you.
Michael.
Yes, just to follow on from that question, difference in antigen presenting cells, dendritic cells between the volar aspect of the arm and the deltoid. Obviously, there's something in it. Do you want to comment?
Yeah, I don't know, Robert, if you want to comment on that. I can talk to you about the preference for where people want it. Do you want to make comment on that question?
Don't have time.
Okay. Well, perhaps we can talk in the coffee break about that, but I can tell you that older people prefer to have it administered. I know, yeah. Yes, yeah. Might talk to Robert about that in the break if that's okay. I apologise. Thank you for your question. Take it on notice.
Please thank Matthew and Cristyn again.